with implications for policy and practice. My name is McPherran Lombardi, and I'm hosting today's call with Sara Mancall from SRCD. Sara will be providing technical support throughout the webinar, so you may see her name in the chat box or hear her jump in as we use the technology. Before we begin the call, I'd like to spend a minute orienting you to GoToWebinar, the technology we are using for the call. All of you should be able to hear the presentation and see the slides on your computer. You can choose to listen by using your computer if you have a microphone and speakers or a headset attached to the computer. Alternatively, you can hear the presentation by calling in separately using a landline or cell phone. All of you should be able to see the PowerPoint slides in the middle of your screen and a box to the right that lists the names of the people on the call. This box may come and go on your screen depending on your settings. If it disappears, you can click on the red tab to bring it back. In the chat box, you have the option of sending a message to me, as well as the option of sending a message to the full group. We encourage you to use this box to answer questions or to ask questions. Over the next few minutes, we also invite you to try out using the chat by introducing yourself to the full group by giving your name and the organization or university you are affiliated with. We also welcome you to use the chat box to ask questions or connect with others on the call. During the presentation, we do ask that you mute your phone line and computer microphone for the purposes of clarity of audio. This webcast will be recorded and is available on the Consortium website with a number of the reports and materials provided by the speakers within the next few days. Before I turn the call over to Dr. Maholmes, who will be giving a short introduction to the topic, I'd like to say a few quick words on the Consortium. As many of you may know, the university-based Child and Family Policy Consortium is a forum of more than 35 individual university-based entities. We have members around the country representing schools, departments, and centers that advance the study of child and family policy. The Consortium regularly hosts monthly webinars like this one, co-organizes the Ziegler Policy Pre-Conference, which is held at SRCD biennial meeting, issues a quarterly newsletter, and runs an active listserv, which helps connect students, staff, and faculty to relevant professional development opportunities and other resources. If your institution is not yet a member of the Consortium, please consider joining. To receive information, please contact our Consortium coordinator, Sara Mancall, who has shared her email address in the chat box. It's now my pleasure to introduce Dr. Maholmes, who is the Chief of the Pediatric Trauma and Critical Illness Branch at the National Institute of Child Health and Human Development. She'll be giving a short introduction to this topic before turning it over to our presenters. Welcome.
Thank you, and good afternoon, everyone. Thank you for joining our webinar today. We're here to delve into the important topic of the biological embedding of child abuse and neglect. And this is a very important topic which we've studied and from the vantage point of the NIH have invested resources to really understand more about this particular public health concern. The SRCD published a social policy report summarizing proceedings from an expert panel meeting convened by the Administration for Children, Youth, and Families, the National Institute on Drug Abuse, and the NICHD, National Institute of Child Health and Human Development, both at the NIH. And the purpose of this meeting was to really help us to understand why maltreatment has such pervasive effects. And the studies on neuroscience of maltreatment have really begun to give us an insight into this, and we wanted to explore in more depth what the underlying mechanisms are of these outcomes for children who have been maltreated. So we brought together a group of experts to look at brain structure and function to help us to understand why victims of maltreatment differ from non-victims with respect to brain structure and function, HPA axis, autonomic nervous system function, epigenetic markers, and so forth. then we wanted to be able to identify risks for poor mental health and physical health outcomes, poor performance by kids, and what all of these factors, how all these factors work together to impact long-term health and outcomes. So we're delighted to be here today to explore this in more depth, to hear from our speakers more research that gives insight on these important topics. We have with us today Dr. Seth Pollak, who's professor of clinical psychology and at Wisconsin, the Wakeman Center of the University of Wisconsin-Madison. He's a professor of College of Letters and Science. He's a distinguished professor of psychology, professor of anthropology, pediatrics, and psychiatry at the university. And his particular interests explore the mechanisms of developmental change. He's interested in questions about the interpersonal, cognitive, and neurobiological mechanisms that are responsible for complex behaviors that children may acquire during infancy, early childhood, and into adolescence. Our other speaker is Dr. Sara Jaffee, who's a developmental psychopathologist in the Department of Psychology at the University of Pennsylvania. She is interested in at-risk families and children and how stressful environments exacerbate underlying genetic vulnerabilities to affect children's development. Dr. Cheryl Boyce will be summarizing and providing a discussion on the presentation today. Dr. Boyce is the Chief of Brain and Behavioral Development Branch at the National Institute on Drug Abuse here at the NIH. She's also the co-chair of the NIH Child Abuse and Neglect Working Group. And we're really looking forward to hearing these presentations. And so with that, I will turn it over to Dr. Pollak to talk about the biological embedding of child abuse and neglect, new insights about developmental mechanisms. Dr. Pollak.
Thank you so much, Valerie. Thank you all for joining in this call, and welcome, and thank you to the Consortium and SRCD for organizing this and inviting me. I'm very pleased to tell you about this recent study and discuss a little bit about how it can stimulate our further thinking about the problems of child abuse and neglect. Next slide, please. Now, I don't need to tell the group that's listening to this presentation about the problems that are associated with early experiences of child abuse and neglect, in fact, in general, early stressful childhood experiences. But it is a good time to remind everybody that it's not only mental health problems that have been associated with these early stressful experiences, but also things that don't strictly fall within the purview of psychopathology. And there are emotion regulatory problems often in these children that disrupt the quality of their interpersonal relationships. There are problems that impact cognitive functioning, having to do with learning and school performance, which affect occupational attainment. And there are long-term implications for physical health. And interestingly, these long-term health manifestations, things like risk for type 2 diabetes and certain forms of cancer and cardiac problems, don't even manifest until the 6th or 7th decade of life. So the problem is that something is happening to people early in life when they are exposed to toxic levels of stress. The problem in understanding this is that it's not one kind of problem. It's affecting multiple domains of development, and it's not necessarily affecting behaviors that we see immediately, but having a cascading effect that can influence an individual across the life course. And so this is the motivation for trying to dig down and understand some developmental mechanisms that could give us clues about prevention and intervention. Next slide, please. We use the term biological embedding, which has become very popular, but I think is very useful, as a way to understand the biobehavioral mechanisms through which experience gets under the skin, the ways in which things that happen to us across different periods of the life course can alter our development in ways that are influencing our health, both mental and physical, our sense of subjective well-being, our learning and the way we approach new information, and our social behavior over the course of our lives. And in my laboratory, what I've been trying to do is steal from non-human animal researchers. Because child abuse is such a messy problem, there are so many covariates, there's so many co-occurring risk factors and cumulative risk factors, that it's just not possible for us to cleanly isolate particular neurodevelopmental mechanisms. But by borrowing or translating from research methods and findings done in more controlled laboratory experiments, we can then try and translate what we've learned from non-human animal studies that have tight experimental control over a family's genetics, over nutrition, over age, over exercise, over diet, and see the degree to which these can help inform our understanding of child abuse. So as I will return at the end of this presentation, we have to be very mindful of the difficulties involved in translating between species. The motivation for the work that I'm going to tell you about is really that, in my view, there are few targeted and effective psychological interventions that are designed for children that have had early stressful life experience. And in my view, we lack the knowledge about specific systems, specific biological, biobehavioral systems that might be underlying these problems and are also amenable to behavioral change. And that's what we're trying to do. Next slide. You see a cartoon here, and it's a father looking down at his new baby and saying, so how do you want to play this? Nature, nurture, or a bit of both? And I enjoy using this slide because what got me into this field was, of course, questions about nature and nurture. Where does human behavior come from? Where does human emotion come from? And it is in many ways an age-old question. What's stunning to me now is that we used to think about inheritance in terms of the letters of the DNA code passed from parents through egg and sperm to their children. But we now know that there's another path, that parental behavior can essentially write information onto the DNA completely, do this completely by bypassing egg and sperm. And in a way, this makes a lot of sense. Think about it this way. Through experience, individuals can use information about the world that they are encountering, the world that they're growing up in, and they can change their DNA to cope with that current environment. It's an environment that we would never know about or have exposure to prenatally. And it adds flexibility to extend some of what we enter the world with that's part of our fixed DNA code. And it's probably very much a mechanism of adaptation and survival. Next slide. Now, when I was in graduate school, we would say, we would have developmental psychology classes, and we would say, is it nature, is it nurture? It didn't matter what it was. It could be social behavior, emotions, language, cognition, causality. We'd say, is it nature, is it nurture? And at the time, the popular answer was to say, oh, it's both. Nature and nurture interact. And it's funny for me to look back on that, because at the time that seemed like such a good, satisfying answer. It's both. Nature and nurture are interacting. But there's a sense in which that's sort of vague. I mean, what's interacting? It's not really answering. That answer never really, in a satisfying way, answered the question about what aspect of experience or social behavior is affecting biology or genes, which genes are responding, how is this all happening? We were sort of doing a little bit of hand-waving. And we're now at a point where we can actually ask more specific questions about what might be going on. Before going on, though, I do want to make one general point. I'm about to talk about new changes in our understanding of genetics and behavior, but I want everyone to understand that biological reductionism isn't going to work either. What we really need to do to understand children's development is work across levels. We need to understand more about the biology of what's in a child's head, but we also need to be able to measure and understand what a child's head is in. And this understanding of what is the social environment, what are the environmental factors that the individual child is encountering, and what are the biological mechanisms that are allowing the child to respond positively or negatively to those environmental circumstances is really going to be the key. In the case of child abuse and neglect, we have some really fascinating research that I love that was done by Sara Jaffee many years ago, showing that the outcomes associated with child abuse can't be explained solely by genetic factors. And so this raises a question about how the experience is affecting a child and what exactly is being affected. Next slide. As we get into understanding what I'm going to talk about, I want to also offer a disclaimer. I am a developmental psychologist. I'm not a geneticist. I've been learning a lot about epigenetics and explaining it and talking to colleagues. I'm hoping I'll be able to answer most of your questions, but there might be a certain level of detail where I don't feel qualified going deeper. The idea behind epigenetics is that genes are not fixed and permanent, that there are changes. And in some sense, the way I like to think of it is there are some genes where it doesn't matter whether you were born with the gene or not, or whether you were born with a variant of the gene or not. What matters is whether the gene is turned on or off. Because if you have a gene and it's not turned on, it's not doing anything. And if you have a gene that does bad things that's turned off, you're probably not going to be at risk. And I've highlighted a couple of things on the slide for you. You'll see there's a red circle at the very top, which is showing that there are many different factors that we now understand can turn on and off different genes. Development, childhood experiences, chemicals, drugs, aging, diet. Not only can characteristics of the environment turn genes on or off, there's now even a suggestion that our interpretations, our subjective perceptions of our environment are enough to flip a gene on or off. And so this actually adds a whole new layer of complexity to help us understand what might be going on. In the case of child abuse and neglect, we know that children growing up in stressful environments overattend to threat and hostility, that this is what they notice in their world. And it could very well, we don't know this yet, I'm speculating, but it could very well be the case that it's the perception of these experiences that's having an effect. There are two different ways in which genes are turned on and off, and I'm going to talk about DNA methylation. This is essentially enzymes that are attached to different parts of the DNA that allow or suppress the DNA to actually create proteins. Next slide, please. The idea behind epigenetics is that alterations to the DNA other than changes to the genes themselves can be passed on with cell division, they can change normal gene expression, they can be caused by early childhood experiences, and they're pretty well studied. Methylation occurs in an active stretch of DNA. I'm going to give another figure to explain this a little bit better, particularly in the promoter region, and it changes the way genes can be expressed. And my thinking about this is that the idea that our experiences, our social experiences, can flip genes on or off may be really helpful for psychologists in thinking about psychological explanations for how experiences can both lead to proximal behavioral problems in early childhood, for example, emotion regulation difficulties, as well as distal problems in adulthood, such as health problems associated with aging. Next slide, please. What we know about this groundbreaking work really comes from work that was done at McGill University a number of years ago by Michael Meany and Moshe Ziff. What they were able to do is take rat pups that were being raised by normal rats that took good care of their offspring, and take rat pups that were being raised by abusive or more precisely neglectful moms and cross-foster them. So a pup born to a good mom was put in the cage and raised by a not good mom, and a pup born to a not good mom was put in a cage and raised by a good mom. And good and bad moms were actually defined in these early studies by licking and grooming, which is caregiving for these rodents, not only hygiene-oriented, but calming and creating kind of security for the rodent pups. Later, when these baby rodents were sacrificed, Meany and Ziff noticed changes to a particular gene, the glucocorticoid receptor gene. And the glucocorticoid system is a system that is responsible for organizing the threat detection system. Glucocorticoids, such as cortisol, play a critical role in the coordination of adaptive responding to stressors in the environment. Next slide. What Meany and Ziff found was very interesting. If you look to the slide on the left, they've graphed separately the responses of rats from high licking and grooming mothers, that's very attentive care, and rats that had low licking and grooming mothers. And what you can see here at the bottom, you see a little dark line. It says pre-S10, S20. S10, S20 is when the rats are exposed to a stressor. And then the rest of the time, P20, P40, P60, P120, is the rat's recovery from having had a stressful experience. What you can see with the lower line is that rat pups that were raised by attentive caregiving mothers showed a normal stress response, and then, but not that high, and then they return to baseline. They reach a state of homeostasis. The rats that received poor caregiving not only show an exaggerated stress response, they have trouble over time coming down and regulating that response to stress. If you look to the slide on the right, or the image on the right, you'll see that they found particular regions of the brain where there were changes in the glucocorticoid receptors. Now, one of the things that I'd like to point out before moving on, and it's a point I've tried to make in previous publications, is that it's not just, oftentimes in child abuse, we focus on deficits. But here, it's important to note that this could actually be very adaptive. We think of anxiety as bad, as being a disadvantage, but in fact, most of us live in low-danger environments. If danger is very high, keeping a low profile, responding quickly to stress may actually be very useful in the short term. And so this is a good reminder for us to think when we see stress-exposed children engaging in behaviors that initially look like pathology, it's sometimes useful to think about what function is being served, the way in which biology might actually be working to promote survival. Next slide. The take-home message from these rodent studies is that better parental care decreases methylation of the glucocorticoid receptor promoter, and that means that there's more glucocorticoid receptors being expressed in the brain. More glucocorticoid receptors means that there's going to be less responsiveness to stress. And so you can see in the silence gene, these little yellow images that I've highlighted are the methyl groups. They are putting a brake on the gene and not allowing the gene to express. When the gene becomes demethylated, these methyl groups disappear. You can see in the activated gene, you just see the DNA. The methyl's gone. The gene doesn't have the brake anymore. It can produce these glucocorticoid receptors, and the system can now work adaptively. Next slide, please. In our study, what we attempted to do as closely as possible is recruit children that had had verified, substantiated cases of child abuse and try and do exactly what Meany and Ziff did with their rodents to see if we could translate the animal findings to human children. And you can see what the group looked like. Next slide. I'll jump right to the findings. And here I'm showing you an image that's a little bit different from the one in the paper because I thought the members of this audience might be interested in it. What you can see here is, look at the middle set of bars where it says CPG site 3. This is a site on the glucocorticoid receptor. The blue line is our controls. These are children from a community sample where there was no record and no indication that we could find of any child abuse. In the green, at the very end, are children where there was substantiated child abuse, and you can see increased methylation on the gene. These are more of the breaks still being on this gene. And what I thought this group might be interested in is the red bar in the middle. These are cases where a Child Protective Service has said to us, we don't have enough evidence to call this a substantiated case, but we're keeping it open. We really suspect there's something going on. We're not closing the case. We just don't have enough evidence. And you can see that when the CPS caseworkers have a strong suspicion that they should continue monitoring, those children are actually looking on this particular measure exactly like the children with the substantiated maltreatment. Next slide. So what I've shown in this next image is a picture of the glucocorticoid receptor gene. What I've highlighted with a circle is the region of the gene that was originally identified by Ziff and Meany as being responsive to parental care in rodents. And what was also shown by them is that changing this particular part of the gene was offsetting the glucocorticoid HPA system, not allowing animals to regulate stress responses. And then I've also highlighted and circled the region of the genes that we found to be responsive to parental care. And you can see there's overlap in CPG sites 3 and 4. And then we found something on CPG site 7. What this means is that abused children are showing more methylation on several sites within exon 1F of the promoter region of this gene. And I also want to point out that site 3, a site found in the rodents and by us in children, is a very critical site. This is where nerve growth factor binds to this gene. And so we don't know a lot about this, but this could actually have huge implications for brain development and brain growth and brain repair, which often happens during deep sleep in humans. Next slide. Let me wrap up by leaving some issues for further thought. The first is that translation across species is important but difficult, and we have to proceed with caution. I've certainly presented some data consistent with the view that human genes can be turned on and off, but it goes without saying that this study cannot infer causality or specificity of processes. Next slide. I also want to point out that animal models are incredibly useful for controlling confounding variables that we cannot control in human studies. But rodents are not children. I mean, I have 2 children. They're 8 and 10 years old now. It's been a while, but I don't ever remember anogenital licking and grooming as part of my parental behavior. And so we have to be cautious about the way we're linking parental behavior in a rodent and how that might map onto things that abused children might experience, being denigrated, having self-confidence undermined, the whole aspect of emotional maltreatment and abuse, which of course is impossible to demonstrate in an animal model. Next slide. The other limitation here is that we cannot take tissue directly from brains the way we can in rodent studies. We have to take it from leukocytes in the blood. And we don't know whether genes taken from white matter in the brain and genes taken from blood in the periphery have the same function. If we want to be really liberal and say, let's assume they're doing exactly the same thing, the implication of the findings we have in children is that there would be fewer glucocorticoid receptors in the brain, and this would lead to impaired stress regulation in children. That would mean there's impaired negative feedback to the HPA axis, and these kids will have a hard time calming down and ramping down negative emotions. But we really can't say that for sure. What we can do is accept a more conservative interpretation. What does it mean for a gene to be methylated when we find it in the blood, in the periphery? What this means is that these children have fewer glucocorticoid receptors on white blood cells. This would lead to higher inflammatory activity and less cortisol regulation, and we would expect these children to have poorer health. Next slide. In fact, there are a number of studies, not only our work, but some work by Andrea Danesi, showing that maltreated children do have higher inflammatory activity. They are sick more, they get ear infections, they get colds. In this case, we found that they were less effective at containing and fighting off viruses. And so the peripheral interpretation really does seem to hold some water. Next slide. As Sara Jaffee is going to talk to you about next, we can really think about a broader framework for understanding what's going on here by looking at the degree of stress in the environment that children experience and how that might or might not affect child development. Next slide. I'll summarize by saying that these data suggest that social experience can alter human physiology, but I also want to note that this does not necessarily suggest that anything permanent is going on. Rodent studies have suggested that epigenetic changes are reversible, and when the environment changes, there's no reason to believe that these genes can similarly not change. And what I'm hoping we can do is discover ways to target and change processes that are affected in these children. This might require personalized or individualized treatments based either on the nature or timing of children's experience or individual differences in children's sensitivity to experience. On the last slide, I've listed our webpage. You can download other papers that I've referenced here, and I thank everyone for their attention.
Thank you, Dr. Pollak. Our next presenter is Dr. Sara Jaffee, who will be presenting on a recent SRCD policy report that she co-authored. Thank you for joining us, Dr. Jaffee.
Well, let me start out by echoing Seth's thanks to the Child and Family Policy Consortium and to SRCD for organizing this call and for inviting me to talk about the policy report. And let me thank Seth for such an interesting and clear presentation on epigenetics. The psychologists and psychiatrists have been studying the adverse effects of abuse and neglect on children's development for a long time now. There are longitudinal studies that span the period from childhood into adulthood showing that abuse and neglect have adverse effects on children's emotional and behavioral health, other life course outcomes. And many of these studies have isolated the unique effects of abuse and neglect from the effects of poverty more generally. Arguably, public attention has been captured more, however, by recent research showing that abuse and neglect have adverse effects on physical health. So in the mid to late 1990s, members of the Kaiser Permanente Health Maintenance Organization in San Diego were asked to complete surveys where they reported retrospectively on adverse childhood experiences, including abuse and neglect, among other things. They were also asked about parental psychopathology, substance use problems, other forms of family violence. And the take-home message of the ACEs study, as it was called, the Adverse Childhood Experiences Study, was that cumulatively these different adversities increased risk for cardiovascular disease, for diabetes, for cancer. And so it suddenly became apparent that abuse and neglect didn't just make children sad or anxious or angry, but it made people sick, and it made them sick in ways that cost taxpayers a lot of money. So in part, as a result of this research, health psychologists and pediatricians have become very interested in the mechanisms that might link experiences of abuse and neglect to poor physical health. They've become interested in the biological embedding of abuse and neglect. And in 2012, the American Academy of Pediatrics published a report that was authored by Jack Shonkoff and Andrew Garner, where they discussed the lifelong effects of exposure to toxic stress and adversity. And they defined toxic stress in this paper as chronic and uncontrollable events resulting in strong, frequent, or prolonged activation of the body's stress response system. And they contrasted this with positive stressors and with tolerable stressors that a child could perhaps manage in conjunction with a supportive adult. And they outlined the effects that exposure to toxic stress might have on the developing brain, on the immune system, and on the neuroendocrine system. Next slide. And so what they reviewed, and in fact, if you could actually advance through a few slides, because there are various circles, and yeah, there we go. Okay. So what they showed is that in the short term, what happens is that potential threats are identified by the amygdala with input from the hippocampus and the prefrontal cortex. And the amygdala triggers firing of the locus coeruleus, which activates the fight or flight response. The sympathetic adrenal medullary system triggers an immune response leading to inflammation. And the amygdala also activates the hypothalamic pituitary adrenal axis. And so it stimulates the release of corticotropin-releasing hormone from the paraventricular nucleus of the hypothalamus. This stimulates the release of adrenocorticotropic hormone from the anterior pituitary, and this finally stimulates release of glucocorticoids, cortisol in humans, from the adrenal cortex. And glucocorticoids feed back on the HPA axis to shut it down when a threat is abated. It feeds back on the immune response when a threat has abated to reduce levels of inflammation. And so in the short term, this is a very adaptive coordinated response. The activation of the sympathetic adrenal medullary system increases blood pressure and heart rate. It diverts blood to muscle, and so it facilitates flight from threat. The HPA axis mobilizes stored energy. The immune response, the inflammation triggered by the immune response, attacks possible pathogens when these threats are physical, for example. The problem is what happens over the longer term with this response. Next slide, please. So over the longer term, chronic activation of the sympathetic adrenal medullary system results in chronically high blood pressure. Chronic activation of the APA axis is associated with obesity and with chronically high or low levels of cortisol. And finally, chronic activation of the immune system results in chronically high levels of inflammation. And in the brain, we see, for example, dendritic atrophy. So over the long term, this coordinated response leads to potential wear and tear on the organism. So our goal in the social policy piece was to review the recent literature on how abuse and neglect get under the skin, affect brain development, affect the development of the HPA axis and the immune system, and to think about some of the policy implications of this work. Next slide, please. There is now a growing body of research, much of which Seth has been involved in producing, in fact, showing that children who have been maltreated have variations in both brain structure and function compared to non-maltreated children. And some of those differences have to do with brain volume. So for instance, you see reductions in the size of prefrontal cortex, reductions in hippocampal volume, areas of the brain that are involved in sort of executive control, involved in memory, among other things. And in the amygdala, a part of the brain that is involved in, among other things, processing of emotions, you see functional differences in response to emotional stimuli. So for instance, in some of Eamon McCrory's work in the UK, he's shown that children who are maltreated show relatively greater activation of the amygdala to angry faces versus neutral faces compared to control children. And some of this is consistent with other literature showing that children who are victims of maltreatment may be hypervigilant to threat, right? It may be that some of these functional differences that we're seeing in the amygdala may actually underlie this hypervigilance that we observe behaviorally in children who've experienced abuse and neglect. Next slide, please. There's also been a great deal of research showing that abuse and neglect are associated with dysregulation of both the HPA axis and the immune system, systems that are tightly linked. So, for example, it's been observed that hypersecretion of corticotropin releasing factor, releasing hormone, could result in glucocorticoid receptor insensitivity, right? So glucocorticoid receptors in the immune system, in the hippocampus, on the hypothalamus, are responsible for shutting down those systems when a threat has abated. And one hypothesis that's found some support is that abuse and neglect are associated with insensitivity of those receptors to glucocorticoid signaling, so that those systems are not shutting down as they should be after a threat has abated. It's also possible that hypersecretion of corticotropin releasing factor is having downstream effects at the level of the pituitary, where receptors on the pituitary are becoming desensitized, resulting in eventually less cortisol being available, sort of lower levels of glucocorticoids being available, again with implications for how the HPA axis and immune system might be shut down once a threat has abated. So there's growing evidence that abuse and neglect are associated with dysregulation of both the HPA axis and the immune system. And there's increasing evidence that some of these forms of dysregulation may be associated with, say, depression, where, for example, glucocorticoid receptor insensitivity is being linked with maintenance of depressive symptoms. Next slide, please. So one of our challenges in producing the social policy report was to think about the policy implications of this research on the biological embedding of abuse and neglect. And one of the things that my co-author, Dr. Cindy Christian, and I concluded is that in some respects, the policy implications aren't really so different from the policy implications of research showing that abuse and neglect have effects on emotions and behavior. But there is a twist to this, in the sense that increasingly this research on the biological embedding of abuse and neglect is suggesting that there are common pathways by which stressors like abuse and neglect are not only affecting emotional and behavioral health, but are also affecting physical health. And one potential implication of this is that developing effective preventive efforts could be a very cost-effective way of targeting multiple physical and mental health outcomes to the extent that these arise from dysregulation in common pathways. Next slide, please. Another implication of this research builds on the fact that, again, abuse and neglect seem to have effects that are very widespread, that not only affect mental health, behavioral health, but also physical health. And this suggests that efforts to both prevent maltreatment and to intervene when maltreatment has occurred are going to require efforts that are coordinated on a number of levels. So one of those levels has to do with formation of multidisciplinary teams of individuals that make contact with children who may be at risk for maltreatment. What you often hear pediatricians say, for example, is that they don't necessarily feel qualified to determine whether a child is a victim of abuse or neglect. They don't have confidence that the child welfare system will actually be able to help a child. They are worried that making a referral to social services may actually do more harm than good. They're worried about getting entangled in court cases, for example, if a family is referred to the child welfare system. And what you often observe for social workers is that they're frequently being asked to make medical decisions about children and about services for children's health that they're not really qualified to make. And so one implication of this is that multidisciplinary teams that bring together pediatricians, social workers, psychologists, psychiatrists, provide good opportunities to look at a child holistically, to look at all of a child's needs in their entirety, to provide different individuals who make contact with that child with support to make the kinds of decisions that they need to make, to support pediatricians in making decisions about which families are at risk, to support social workers in making medical decisions for children, for example. And finally, to be able to monitor, for example, the use of psychotropic medications by children who are in the child welfare system. We know, for instance, that children in the foster care system, for example, have very high rates of psychotropic medication use, and that medication use isn't really being effectively monitored. But that could happen in a more effective way with multidisciplinary teams. Another way that efforts to protect children and promote their well-being could be coordinated would be through greater resources devoted to the development of electronic medical health passports. So the problem for many children in the child welfare system is that in making multiple residential transitions, they lack documentation regarding their health, regarding other aspects of their backgrounds. And that a system that could coordinate paper and electronic records could go a long way to facilitating medical services and social services for children who are making multiple residential transitions while in the foster care system. Next slide, please. And finally, although we know that there are some interventions that are effective in treating the depression and the anxiety and the symptoms of PTSD and the aggression that tend to be associated with children's experiences of abuse and neglect, interventions like trauma-focused cognitive behavioral therapy, for example, we don't have a great handle on why those interventions work or for whom they work best. And the research on the biological embedding of abuse and neglect potentially provides mutually beneficial opportunities for intervention researchers to get a better handle on why their interventions are working, right, to the extent that interventions are reversing the effects of some of the dysregulation that one might observe in the HPA axis, in the immune system, in brain function, for example. And it might benefit basic researchers by providing some support to hypotheses about the biological underpinnings of psychopathology and other physical health problems, to the extent that, again, interventions are improving symptoms of depression or symptoms of PTSD, and that they're doing so because of the effects that they're having on some of these biological systems, that provides stronger evidence that those biological systems are indeed likely to be causes of some of the mental health problems and physical health problems that we typically observe in children who are victims of abuse or neglect. Next slide, please. This research leaves us with a lot of unanswered questions. So, for example, although there are now a growing number of studies showing that abuse and neglect are associated with alterations in brain structure and function, in the HPA axis, and the immune system, it's not always clear whether those dysregulation in those systems is actually associated with psychopathology or with physical health problems. So there's some work starting to show those links, but much more of it is needed to show that the biological embedding of maltreatment is in fact what is underlying physical, emotional, and behavioral health problems. It's not always clear why some of us are more susceptible to experiences like abuse and neglect than others of us. So again, there's a growing literature on how genetic variation may explain some of those individual differences in susceptibility, but much more work is needed in this area. And finally, from a developmental perspective, it's not entirely clear when we're most susceptible, whether the timing of an adverse experience matters more than the sheer chronicity of that experience, or whether there are particular windows in which the effects of adverse experiences like abuse and neglect are reversible. So there is a lot of work left to do, but fortunately there are a lot of good people working on these questions, and it's an exciting time to be looking at this research. Thank you.
Great, thank you, Dr. Jaffee. We're now going to have a few cocooning remarks by Dr. Boyce.
Well, good afternoon. It's not much more I can say after two great presenters who as well as my colleague, Valerie Maholmes, who co-chairs the NIH Child Abuse and Neglect Working Group. We both have had the pleasure of working with two outstanding grantees in this area who not only have an understanding of the biological underpinnings of child abuse and neglect, but also have a very savvy understanding of what that means for policy and practice, and that is what this session was about here today. It's not so long ago, actually, that child maltreatment was even recognized as a syndrome, as a problem. And since then, as child maltreatment has been identified in child welfare, we've also had the expansion of knowledge in terms of biology, neuroscience, psychosocial understanding factors, and the advancement of child welfare systems and knowledge on family, child development. So we've had these big understandings grow and not always complementing each other. So it's important now that we continue to put what we know about biology and child development, as well as what we know in terms of our best understandings of what's going on in families and policy and practice together so that we can really improve the lives of children. I was struck by a couple of the comments from both the speakers. One, especially by Dr. Pollak, who I've known for a long while, and he has looked at several large cohorts of children who've been exposed to all types of circumstances and stress and trauma. And to think about it from the understanding in terms of epigenetics that there's something that can be turned on, that there may be something there, but whether or not we turn it on or can we turn it back off, and what we can do, which allows for us to understand that there is a place that we can think about for prevention, and there's places that we can think about for intervention, which also gives us hope. And then also, I'm struck by some of the comments from a dear colleague, Dr. Jaffee, who also has really done some of the seminal work in this area, again, really emphasizing the interplay of what also goes on in the environment, and that while there's some biology embedded, it's the interplay of what we sometimes talk about here at NIH as not just genes by environment, but genes by environment, that there are these dynamic processes that really impact the course of a child's life and really can impact what can happen in terms of prevention or intervention for child abuse, and should really apply to how we think about future policy and practice. So we don't have that much time, and there may be a few questions, and I know some of the ones that we had been considering early on were quite thoughtful, and I want to allow time for our speakers and my colleague, Dr. Maholmes, if she has any added comments as well. Here at the National Institutes of Health, we have been longtime supporters of really increasing the research base and innovation to inform policy and practice, along with our colleagues at many of the other agencies within Health and Human Services, and including CDC, as well as thinking about how this fits into all aspects of development and other areas of the child's life. So with that, I'll allow Dr. Maholmes, if she has any comments, to give maybe some closing words, and then turn it back over to our leader and any questions or comments that we can take to the speakers as well?
Not a whole lot more to add. I'm very encouraged by the two presentations, and especially connecting the science to the policy is really, really important. And from our perspective here, we want to fund the best science, and we hope that that science will give rise to policy and to informing policy. And we would encourage you to continue to work collaboratively, and a lot of our efforts now, and I suspect in the future, will be to encourage those multidisciplinary teams, as Dr. Jaffee talked about, to really kind of look at this major public health concern through the prism of multidisciplinarity, and I think that's the only way that we're going to be able to get at the heart of some of these questions. So with that, I'll stop and let others ask their questions.
Great. This is Caitlin. I'm going to try to combine a couple questions with our remaining 5 minutes. All of the speakers today did a fantastic job in communicating a very complex issue, and several of our callers were sort of interested in how we can do a better job and really sort of in communicating this research to the public, both just to the public at large, but then also to professionals and practitioners who work with children and families. There's so many different sort of facets that you explained very well and simply today. How can this research be translated in a way that can be well understood and used, particularly by those who are really working directly with children?
I think that one important way that we can ensure that this new research, particularly as we're branching into new areas where we have little of a framework for understanding them, but yet we're finding interesting new nuggets, is making sure that we communicate issues about context. What do the findings really mean? But also, when we point to something that might initially look like a deficit or problem, encouraging an understanding that frames it within the social and emotional context that the child is developing within. Maybe something that's creating problems with peers on the playground in 3rd grade was actually something that makes a lot of sense and was very adaptive in infancy. And so we need to look at the big picture rather than, say, generating a laundry list of deficits and symptoms and problems, because that understanding will let us see sort of how biology makes sense. The other thing is biology isn't destiny. Biology doesn't mean fixed. And it's sometimes the general public will feel like if we talk about something as being genetic or hormonal or in the brain, it means someone is like that. They've always been like that. They always will be like that. But there's a tremendous amount of research about flexibility, reversibility, sensitivity of biological systems to the environment. And it'd be great to get more of that work out there, too.
Great. And we also had another question that might be interesting to a number of the participants. And that question is, what are your suggestions as researchers for other researchers, particularly in developmental psychology, who want to include more physiological markers in their research? What sort of recommendations do you have as a researcher to another researcher?
Well, there are a lot of opportunities to become educated in how you can incorporate biological markers into your research. There are workshops, for instance, that Northwestern University offers in the summer. There's a summer institute, a biomarker institute that's offered. The Institute of Psychiatry in London offers a genetics training summer school program. The Institute for Behavioral Genetics in Boulder, Colorado offers workshops in both behavioral and molecular genetics. So there are those sort of formal short courses that one can do. You know, another thing is to talk to colleagues in your department or in neighboring departments who maybe use some of those methods. They may not work on exactly the same questions you do, but they might work on questions that are close enough that you can have a productive discussion about how the methods they're using can be brought to bear on the kinds of questions that you're interested in asking. And finally, there are companies like Salometrics, for example, that can do a lot of the lab work that, many of us who are trained in developmental psychology are not trained to do ourselves. So, making use of some of those resources for the assays, for example, and similar sort of resources exist for genotyping. Making use of those can sort of take that piece of the research out of your hands and hand it over to someone who will do a really reliable job with that part of it.
Thank you, Dr. Jaffee. It's 1 o'clock now, so I'm going to wrap up the webinar in the interest of time. I wanted to thank all 4 of our presenters today and also to our participants. This is actually the largest call ever hosted by the consortium. Nearly 150 people have been on the line. So we want to thank everyone for joining us, for these really interesting presentations, and for the interesting questions posed as well. If you or your organization has questions about joining the consortium, please email Sarah Mancall. Her email is on your screen right now. And we appreciate everyone's participation today. Thank you.